Yeah, so thanks for the question. So Lexeo is a in vivo gene therapy company. We use adeno-associated viruses to deliver the therapeutic gene of interest. We are a company today primarily focused on genetic cardiovascular diseases and central nervous system diseases. Within the central nervous system disease space, we're focused specifically on several indications associated with Alzheimer's disease and an ultra-rare lysosomal storage disease. On the genetic cardiovascular side, we're focused on indications like Friedreich's ataxia and arrhythmogenic right ventricular cardiomyopathy. In general, we tend to focus on larger rare diseases or more prevalent diseases such as Alzheimer's disease. And in general, we also tend to use a similar technology platform across the programs that we're interested in. So it's been a very exciting journey for the company over the last couple of years, and we're excited to potentially deliver very great treatments for patients in the future. Yeah, so the company's origin actually is a spin out from the genetic medicine lab at Weill Cornell, so the Cornell Medical College here in New York City. And the scientific founder of the company is Ron Crystal, who is one of the early pioneers in the gene therapy field. He was involved with dosing the first patients with gene therapy utilizing a virus in the early 90s. And he's been involved in several other milestones in the field since then. He also happens to be the chair of the Department of Genetic Medicine at Cornell. So this was the place where the foundational science for Lexeo was sourced from. As we progressed as a company, we also acquired another gene therapy company. This company was focused on genetic cardiovascular diseases. And this company was created from the labs at UC San Diego, from the cardiovascular labs there, from Professor Eric Adler and Farah Sheikh, both of which are, I say, relatively prominent in the cardiovascular gene therapy space. Eric was the inventor of the existing gene therapy program for Danon's disease that another company is developing today. So we have some very strong ongoing collaborations with both Cornell and UC San Diego, and they are, a very important part of our, research ecosystem. Yeah, so I think there's a pretty meaningful difference between the types of indications you can treat effectively with gene therapy in the central nervous system. And in particular, I think that the challenge that you're describing is typically more so the case with intracellular proteins, where you do not see the cross correction of each cell, meaning you have to transduce every single cell in order to see an effect in that given disease. In our central nervous system pipeline, we're typically focused on secreted proteins, which means you can transduce a handful of the cells, they express the protein, and therefore you can have a therapeutic effect in that manner. So really, in a way, it doesn't matter that you're transducing every single cell. You just have to transduce a sufficient number such that you're expressing a sufficient amount of the protein. So this is where we're focused in CNS, is entirely on secreted proteins, for which CLN2 and APOE4 Alzheimer's disease, represent, that profile. Importantly, I would start with the cardiovascular data, because I think this is, for these programs, relatively transformational data that's been being presented. I would first start with our cardiac, Friedreich's ataxia program. And just to give some background on the disease, this is typically known as a neurologic disease for patients that, begins in childhood. These are patients that are 5, 8, up to 10 years old when they first, see the onset of the disease. This typically progresses until they reach adulthood, when a form of cardiomyopathy associated with the disease emerges. And it's this cardiac disease that's actually the cause of death for up to 70% of Friedreich's ataxia patients. So for that reason and some others related to just the amenability of the cardiac target to gene therapy, we decided to focus on the cardiac pathology of Friedreich's ataxia first. So the abstract and the presentation that around our Friedreich's ataxia program that's part of ASGCT is really laying out almost all of our preclinical data supporting that program, everything ranging from murine efficacy studies in 2 different disease models to non-human primate studies that looked at biodistribution and safety, and really lays the groundwork for the start of the clinical study that we're expecting to launch within 2022. This was also the foundational scientific work that was part of the IND that we had recently cleared by the FDA associated with that program. So, we're really excited about the possibility of bringing the first gene therapy for Friedreich's ataxia to patients. I think the patient community is very excited as well. So there'll be, I think more to come on this program and more data in our future. The second cardiac program where we have a poster at ASGCT is for arrhythmogenic right ventricular cardiomyopathy. So this is another genetic cardiovascular disease. It's a disease of very high unmet need, similar to Friedreich's ataxia. There's no existing therapy. Typically this disease of arrhythmias can in some cases result in sudden death for patients that are during exercise or other activities. So as you can see, obviously the unmet need there is, very significant. We have completed a set of, preclinical proof of concept studies in partnership with our colleagues at UC San Diego. And these have effectively demonstrated an ability to almost completely reverse the disease pathology in the disease model mice. And then when dosed early, we can have the mice with almost a survival that's very similar to the wild type mice. So this is also, I'd say, foundational data for this program, really demonstrating a pathway to a clinical candidate for us, and then ultimately through to IND enabling studies. And that's, where we're headed with this program. There's some other abstracts there. We have one in relation to one of our central nervous system programs. As I discussed, we're focused on a subpopulation of Alzheimer's patients that have the APOE4 gene. So these are APOE4 homozygotes, which confers the highest genetic risk of developing Alzheimer's disease. And we have a currently ongoing phase 1/2 clinical study in APOE4 homozygotes, where we're delivering the APOE2 gene, which is seen to be protective against developing Alzheimer's disease. We're using the APOE2 gene as a therapeutic. But we have 2 other preclinical approaches, one of which is being highlighted at ASGCT, where we're delivering the APOE2 gene, but we're simultaneously suppressing the expression of the APOE4 gene. So this should see an even more dramatic conversion of the APOE4 homozygote CNS to the E2, E4 heterozygote profile that we're seeking to, replicate the sort of protection that's conferred naturally to E2, E4 heterozygotes. So there's another, abstract that we have at ASGCT related to that program, in effect delivering, showing some of the data that is associated with our ability to suppress the expression of E4 while simultaneously delivering the E2 gene. I think I have and I would agree it's not objective view that, gene therapy has the ability to fundamentally change healthcare. Gene therapy in most cases is a single administration, functional cure to the disease that it's targeting. So if we're talking about, cures for diseases over time, this can, dramatically reduce the healthcare costs, across, every healthcare system, around the world. But in a way, gene therapy's greatest benefit is also one of its greatest challenges, is that because it's a single administration therapy, you have to find exactly the right dose because you have only one shot at, at treating the patient. So I think this is the most significant challenge that, we're facing now in gene therapy, is that, there's been a lot of great preclinical work that's been done. A lot of it's been done in, the academic setting. But as we take that work into the clinic and actually translate it into clinical trials, I think there's a lot that we're learning about gene therapy, both in terms of potentially, the limiting dose in gene therapy, some of the side effect profile that, we're beginning to see in the clinical setting. But I think we're learning a lot through this process, both the companies, the FDA, the academic community, et cetera. And I think the gene therapy sector will work its way through these challenges with these learnings. And what this will open the door to is a therapeutic modality that has the potential to, cure hundreds of diseases out there. And I think we'll start with seeing cures to a lot of the genetic diseases. But then over time, I think we'll see gene therapy applied also to acquired diseases as well, to much larger diseases over time. So I think we're at the tip of the iceberg today. We've seen the spinal muscular atrophy gene therapy, Zolgensma, have a very significant positive impact on patients. I think we'll see dozens of other therapies of that profile coming to market in the coming years. And if you look down the road in 10 years, I would expect that the top 10, pharmaceutical products are either, gene therapy or gene editing products that are focused on, treating and curing some of the world's most challenging, genetic and acquired diseases. And so that's kind of where we see gene therapy going over time. And we think the field will continue to grow, in line with that. I think delivery is another challenge. I frankly would put the challenge of finding the right dose is, maybe neck and neck with that one. But I think there's two sides of delivery. So you think of gene therapy as being for, central nervous system diseases. So you're talking about central nervous system routes of administration. And as we were discussing earlier, one of the challenges in CNS diseases is to treat these intracellular proteins and assuring, sufficient biodistribution to treat that given disease. And so I think you really need to look at as very much an indication specific, challenge. I think there's some indications where you need to get to very specific areas of the brain in order to treat the disease, and there's a certain route of administration required for that. There's others where you need to bathe the brain in vector in order to treat the disease, and there's other routes of administration that are appropriate there. So I don't think it's a one-size-fits-all question on delivery from a central nervous system disease perspective, and it needs to be very much indication and disease specific. Now, if we jump to what we call systemically administered gene therapy, I think that presents a different challenge in that you need to find the right vector and the right construct that can yield a tropism profile or an affinity to a certain organ that will ultimately allow you to cure the disease, but will not create significant off-target effects. And that's the challenge of systemically administered gene therapy, is that you need to deliver a significant amount of vector into the human body, but you need to ensure that it's going to the right places in the right amounts. And so that's another question of delivery. But I think as we've seen at ASGCT this year, there's been a lot of developments in respect to novel capsids and novel expression systems that are meant to be very much organ tropic, so focused on specific organs. And what this should allow is an ability for the gene therapy field to progressively reduce the systemic doses that are being delivered to patients, while not limiting the efficacy that patients can benefit from. So what the long and short of it is, we should see less off-target effects, but we should see the same or even greater efficacy through some of the novel capsid platforms that we see emerging. And Lexeo as well has pretty significant novel capsid efforts ongoing. Today we're focused mainly on the cardiovascular side of things, but we're also doing some work on the central nervous system side in respect to novel capsids as well. I think we're seeing a lot of movement on that front. I mean, the first thing I would separate is the costs of manufacturing between ex vivo gene editing and in vivo gene editing. I think we're talking about different orders of magnitude with respect to that. Lexeo is an in vivo gene editing, gene therapy company, and that's where our CMC platform is been focused. But even within in vivo gene therapy, we have seen some challenges with costs there. And the reason for that is a lot of the gene therapy programs that companies ultimately license in or take on have originated from the academic setting, and typically in the academic setting they're less focused on finding the most efficient cell lines to use for manufacturing. They want to find a cell line that's flexible enough that can accommodate different discovery research constructs that they're attempting in animal models. So oftentimes when companies do bring those academic programs in and they attempt to scale them, they have a very difficult decision that they're faced with of, do they change the cell line to one that has more, let's call it scalability, that could be more efficient over time from a commercial perspective, but then you run the risk that you're not able to achieve the same results with that new cell line that you achieved in the academic setting, or do you continue with the academic manufacturing process and cell line, therefore eliminating the risk that you will see different results, but therefore it's less scalable over the long term. So I think that what I would expect over time is to see more companies transitioning their manufacturing platforms from the legacy academic processes, from the legacy academic cell lines to cell lines and processes that are more commercially efficient, more scalable. And I think as we see companies transition to those processes and cell lines, we'll begin to see the manufacturing costs reduce over time. I think it'll just take the industry some time to come to that realization that efficiency in manufacturing, scale in manufacturing may not be achieved via some of the academic manufacturing processes and cell lines that are out there today.